Hello everyone, my name is Hannah Gill, and this summer I worked at Auburn University studying the cryopreservation of blue catfish sperm. Today I will be giving you a tour of my lab and telling you a little bit more about my project. The first stop on our tour is the EW Shell Fisheries Center ponds. Here's where we house our female channel catfish and male blue catfish. We have these two species because they are often hybridized together in the aquaculture industry to produce food for people. When we are ready to perform an experiment, we use big seines to catch the fish and take them out of the ponds and move them into these big tanks in the counting shed. One of the issues with reproducing hybrid catfish is that male blue catfish take a long time to sexually mature, and once mature, the only way we can collect sperm from them is by euthanizing them. Here I am dissecting the testes of a male. Now we move into the dry lab. Here, I am cutting up the testes to extract the sperm from the male. Once extracted, we then dilute the sperm to make sure we have the same amount of cells in each sample. Then we are ready to cryopreserve. We use the methanol shown here to keep the cells from being damaged when they are frozen. We then store the sperm in cryo straws. Here are 0.5 ml straws. These are usually used to store sperm in and develop cryopreservation protocols with. But for my study, I experimented with putting the catfish sperm in 2.5 ml straws. I'm experimenting with using these bigger straws to make it easier for farmers to fertilize large batches of eggs at a time with cryopreserved sperm. Once the straws are filled, we then put them in this device called the control rate freezer. This device uses liquid nitrogen to chill the straws to below -80 degrees Celsius. Once frozen, the straws are stored here in the liquid nitrogen dewar. We then use a water bath to thaw the straws, and we are able to look at their quality after they've been cryopreserved using CASA, or computer-assisted sperm analysis. This computer software allows us to look at the sperm's motility, how many are moving, and velocity, how fast they're moving. The last stop on our tour is the hatchery. During the spawning season, we collect the eggs of female channel catfish and fertilize them with the sperm of male blue catfish. The eggs are stored here in these baskets within the hatchery's tanks. This summer, in addition to my project, I got to help my grad student with her study, comparing the offspring of eggs fertilized with cryopreserved sperm versus those fertilized with fresh sperm. This research will see if there are any deformities in the cryopreserved offspring compared to the fresh offspring, and help farmers have a better understanding that they can use cryopreserved technology to fertilize their eggs without bad outcomes. Working with cryopreservation has been such a cool experience, and through my study I hope to help improve the ease of using cryopreserved sperm for catfish farmers.